All right, so this is molecular biology crash course. So this is pretty important information to know before we jump directly into cancer. Tika, you can also take your call outside if you want to. And this has some roots in O-level biology, not that much. I'm assuming, has everyone here taken O-level biology? Okay, then we're good. So it's just going to be basic over here, except we're going to take a little twist. It's a little jump scare where I'm going to introduce some new concepts that are more important for our research purposes. If I can just scroll through here without any interruptions. Okay, so the core idea of our topic is the central dogma. It's a theory that describes the expression of genes. So whenever we talk about expression of genes, it contains three steps, or one thing to note, and then two steps, which is the genetic information. Genetic information is encoded in DNA. Tika, it's transcribed to RNA, and then it's translated into a protein. And one thing to note is just the DNA is non-functional. The proteins are our ultimate goals. So that's where we're going to be focusing on because that's the main mode of cellular function. Tika? This is a biological circuit, okay? It just shows all the like interactions between different proteins. So like when we're looking over here, I'll just zoom in. Every circle represents a protein, so it's just showing how different proteins interact with each other. Like suppose this protein turns on this protein, and that protein turns on another protein, right? So we call that a protein cascade. You can think of it like a domino effect. And as you guys can see, like all of these arrows converge over here where it says changes in gene expression. So the ultimate goal of these protein pathways is to cause a change in gene expression so that different genes are expressed, and then our cellular function, our cellular activity changes. Tika? And you guys can stop me anytime if you guys need to explain anything else. This is pretty, interactive, hopefully. And then keywords to remember, since we're doing it in terms of cancer, it's going to be like motility, which is cell movement, proliferation, which is cell division, and then cytostasis, which is inhibition of cell division. Okay, now we're going to move on to gene expression. It's again just like how we talked about in central dogma. It's like gene is transcribed to mRNA and then translated into a protein. And then one thing to note is that some genes are master regulators, so they can control other genes as well, like tens, thousands, hundreds other genes. And then we get to the concept of differential expression. Differential expression basically just means that we know that every cell in our body has the same DNA, but how are they acting differently? This is because some genes are more expressed in certain cell types, in certain cases, like different stimuli as well, Tika, and then at particular times. So that's the difference in the cellular activity. Like every cell will have the same genes, but this is how they act different. And then related to your research, you guys are going to see a lot of this type of application of this, which is in a control group. We are applying a drug which stops the transcription. This arrow represents transcription. We're applying a drug to the cells. We get a big sample of cells, and we apply a drug that stops the transcription of a gene. And then in the treatment group, in the control group, it's regularly not transcribed. Basically, it can go vice versa as well. What we're doing is we're trying to see the difference in the cell activity when it's expressed versus when it's not expressed. So this can help us like identify what genes are related to what characteristics, what function of the cell. And then in terms of cancer, we can identify cancer causing genes, understand tumor behaviors, and then develop new treatments. So one example of this was like one of the research papers I cited in my research project was like they applied 5-AZA-DC, don't need to remember it, but it's a drug to glioma cells, which is brain cancer cells. And they saw that it was more expressed when the drug was applied. So basically, we were able to guess how the gene was not being expressed in cancer cells, and then we're able to develop treatments off of that. So we need to figure out how it's stopping it so that we can develop treatments. All right. Basic code. Just wanted to show you guys some codons and that code for amino acids. Codons is in mRNA. Okay, triplets are of DNA. All right. And as you guys will see over here, this is like the same thing we study in AS, but we study it in a table form. This is just a pi form, so you see like G, and then you go on to which one is which one is in second position as well. Okay. As you guys can see, that there are certain amino acids that are coded by multiple codons or multiple triplets if we push it back, because you guys know that triplets goes to codons, it goes to amino acids. All right, let's talk about mutations. So mutations can be silent, subtle, or profound. Silent basically means that, just like how we saw back here, I might change one of the codons in the mRNA with the change in the gene. TK mutations happen in the DNA. I might change one of the codons, but the same amino acid is being formed. So I can't tell that there's a mutation. So that's silent. Then we go to subtle. Subtle basically means that there is a change in the amino acids, but the characteristics of both of these amino acids, the one that was replaced and the one that is replacing, they have the same general characteristics, so that it doesn't have that much effect on the protein's functions or stability. And then profound is the extreme side. Profound basically just means that it is a big difference. Like we think about sickle cell anemia, which we learned in O levels. The replacement decreases the solubility of red blood cells. Tika. So that's just that. So they can be the direct cause of disease. They can determine the natural course of infection, and they can affect your response to common drugs. So when I was studying this was pretty interesting. I remember my teacher saying that it also like saved her mother's life, because she had sequenced her whole genome, and she figured out that if the hospital had given her a certain medication, then she would react in a very bad way. So that's how we have different responses to certain medications. Sometimes we need stronger ones, sometimes we need different ones. And then I'm just going to go through 3 basic types of mutations. There are more, but there's no need. It doesn't like affect it's not in context to cancer, basically. So it's substitution. I think we all learned this, right? Yeah, substitution, where we're just replacing the C over here with a U, Tika, and then there's insertion. We're adding an A in between the C and the T, and then deletion, where we just take out the T entirely. So, what do you guys know what they call the insertion and deletion under what category of mutation is it? Starts with an F. Frame shift. Frame shift mutation. Yeah, so that's basically it. So when we cut out one or we add one, it ruins everything else, right? And so the protein becomes non-functional or incomplete, whatever. Okay, and then we just go on to basically all mutations aren't equal. Tika, some have more drastic effects than others. Some affect DNA repair enzymes, so they can lead to many new mutations. Some mutations affect master regulators, so that they're going to affect many other genes because the master regulator isn't, transcribed. And then some mutations in proteins that regulate cell death. That's a common one in cancer. And mutations in proteins that determine the state of differentiation. Tika? So this is where we get into the nitty gritty. This is where you guys should, really try to understand. You can always ask questions as well. So whenever you pull up the structure of your gene, it's going to come out like this, okay? It's going to come in a line, basically. The online tools do a really good job of sectioning which parts are which because of what data we know already. So this is going to be your gene. Suppose this is your gene as well. This is your gene. It starts from the 5. This apostrophe means prime. Tika? So there's 5 prime to 3 prime. And you guys should know that we transcribe from 5 prime to 3 prime. All right? And when we look over here upstream, this term, there's enhancers, there's promoters. We're going to talk about all 3 of these. And then whenever we talk about upstream, we're talking about components that are coming before the gene, like upstream. Like suppose if you're waiting at a certain part of a river, upstream is going to be way before, and then downstream is going to be way back here, so after the gene. Tika? And then we're also going to talk about this, the initial transcript and the final mRNA as well. This is a more, this structure should be in your guys' mind, because this is just a more in-depth structure. So we're seeing over here, Aju, no problem. Okay, so we're seeing over here, there's enhancers, just like how we saw back here, right? There's the upstream enhancer. And then DNA bending protein, transcription factors. So the main thing you guys should know is enhancer, transcription factors. We're also going to go through that. Promoter, and then you guys know that RNA polymerase is the one that's helping the transcription. Tika, when we're explaining enhancers, we use the word distal. The distal distance, basically, because usually enhancers are like 1,000 base pairs before the gene, right? They're upstream, right? So their job is basically to recruit all of these proteins, transcription factors. They recruit them, and the DNA bending protein makes sure that they have a physical connection. So these transcription factors form a bridge, and they attach to the promoter region. TK, so they basically just enhance. They enhance transcription. They help RNA polymerase, and we're gonna go through that right now. All right, so let's go through the promoters. It's located upstream, right? So it's located before the gene. It's a landing pad for RNA polymerase to bind to, and there's different types of promoters as well. Like we talked about differential expression, this is what we're talking about. So there's constitutive; these are always highly expressed. There are cell type specific. It depends on which cell it is in, which cell it's present in. And then there are some that can be regulated off and on. So this is the master regulator part, where we can turn on the promoters, and that's how the transcription happens. So basically you can think of it like a door, right? The door to the gene. If we keep the door closed, the gene we can't reach what's inside the room. Basically, if we open the door, then we're going to go through and reach the gene. And then a common thing in most promoters, oh, I can just zoom in like this, is the TADA box, TCAT. It's just thymine, adenine, thymine, adenine. It's just a common part of the promoter, and that's why we call it the TADA boxes with the sequence. Okay. And then we're going to talk about transcription factors. These are proteins that are involved in regulating transcription. So we talked about like these ones that are standing in the middle, right over here, right? And it's a highly specific binding. So when the enhancer, suppose the enhancer causes one to come over here, or recruits it, or the promoter does, Tika, and this is a very highly specific binding, like enzyme and substrate, basically. So one thing you'll see in and generally present on promoters to aid RNA polymerase. So one thing you'll see in some cancers is that a mutant particle is going to come, like suppose he just attached to the promoter region. A mutant particle is going to come in between over here, and it's going to bind instead, right? Just like how we talk about competitive inhibitors, right? It's going to bind instead so that this coactivator cannot bind. And that's how the cancer makes sure that RNA polymerase cannot bind, because it needs these proteins basically to guide it. Tika, does that make sense? Okay. You guys can like talk too, Okay, a mutation can dysregulate a gene or protein. Like a mutant gene means mutant protein. Tika, we talked about incomplete or dysfunctional proteins. Normal gene expressed at the wrong time or place, that's by promoters. Tika, mRNA not translated protein, sometimes they get degraded too fast before they complete their function, or they don't get degraded when they're supposed to be. Or protein is modified in an abnormal way, that also comes under mutant protein. So whether indirectly or directly, the cause of gene protein dysregulation is almost always a mutation. And if I go back right over here, degradation, like cleavage. Remember when we talked about cleavage, like in the metastasis one, like they're supposed to be broken down. and they're not broken down, like, yeah, digested, basically. That, because, yeah, it happens by the same thing, because lysosomes, but you don't need to go into that. Yes, like, we're supposed to break it down, programmed breakdown, basically, right? And then if we go back over here, the thing is that there are like 10,000 to almost a million mutations that occur in each human cell per day. Tika, so like Obed was talking about, the way that our body reacts, or the way that our body regulates this, make sure that we're not filled with cancers, is that apoptosis. Apoptosis is programmed cell death. Wait, I forgot to give you guys the notes. Do you guys have the notes for? My bad. Sound like, I mean, this will help you guys. Yeah, you're right. You're right. I forgot. I was too busy with the screen recording. So these are the same ones. These are the same ones. So if you've already grabbed it, then you won't need it again. Do you all three need it? Just you? It's just like the layout, basically. Again, like, this is not for you guys to rut down necessarily, but like understand it so we can deepen your research. And so what they do is apoptosis to deal with mutated cells. There are DNA repair checks. You guys talked about it in the mitosis chapter. To get repair checks, if it's not repairable, then it immediately dies. And obviously we have like millions of cells, so like it's not like an issue if one dies, or a couple hundred die. All right. And if we go over here, I forgot to mention that when we talk about like regulated off and on, these are like by, like we talked about stimulus, right? When we talk about promoters in the previous part, right? So certain stimulus, like if you see over here, like insulin, we know that that's secreted when we have the fight or flight response. You see a dog and you're running, right? So whenever we talk about like insulin response element, so the response element is just the transcription factor binding site, so that becomes exposed when, and then they recruit each other, like we talked about right over here. They recruit each other, transcription factors recruit each other, and that's how the gene is transcribed. Okay. And then we get down to mRNA splicing. Have you guys covered mRNA in your biology? Yes. Okay, great. Okay, obviously we form mRNA from DNA from certain genes, Tika. And when we talk about mRNA in depth, we name it, we call certain parts exons and we call certain parts introns. What I used when I was studying AS was a acronym called NICE. So non-coding introns, introns are non-coding, and exons are coding. Tika? So our job is, we don't really want the introns because they're not really coding for anything. Tika? That doesn't mean that we take all of them out at once, but what this leads us to is the concept of alternative splicing. So we can form different mRNA transcripts from the same original mRNA by cutting out certain introns. Like if we take out the first and the second, or we take out the third and the fourth, and we leave the first and the second. Tika? And there's an average of 8.8 exons per gene. So this is an example of pro-apoptotic BIM. It's a apoptotic protein. It's involved in apoptosis, like we talked about, programmed cell death. It plays a crucial role in apoptosis. So there's three types of it, basically. Three main types. So BIM-EL, it's the largest isoform. This is where all of the exons are included. So we're not taking out anything from the initial transcript. And then BIM-L, which is we only take out the exon, the third exon, and then there's BIM-S, which is we take out 3 out of 4 of the exons. So, and this causes a great difference in their potency, because BIM-S is the most potent inducer while it's shorter, and then BIM-EL is the longest one and it's the least potent, basically. So I put this little meme over here. I always have to point it out. BIM-S is like the real Reaper and, Simpson character. Um, Homer Simpson. I honestly never watched that show as a child. Is it like for adults? Is it like one of those like adult cartoons? Is it not? Oh, okay. Because I know like Rick and Morty is, and that was popular with kids. Huh? Yeah, Family Guy. Okay. So yeah, one thing I was gonna mention over here, but I completely forgot. No, I think that's it. Okay. So yeah, we can tell that, and then we know that we're we're forming different proteins from this, right? So that's the difference: the protein's functionality, the protein stability. Now let's move on to proteins in depth. Tika, just one thing to again reiterate is that DNA is it's not it's not unimportant, but it's not functional. It's our basically our template. So you can think about it like when we're talking about a recipe book and a chef. So a chef uses the recipe book to make cakes. The cell uses DNA to make protein. So the recipe book is important, but it's not the cake. You're not gonna be eating the recipe book. You're gonna be eating what you make from it, right? And then RNA, it's usually single stranded. It's the intermediate, and there are many different types of RNA. Like there's tRNA, some of them have like catalytic activity as well. So because there's many different types of it, it's a little bit more functional. Like we talk about tRNA. tRNA is functional because it brings in the amino acids to the ribosome. You guys have gone through that, right? Okay, great. So yeah, so it's a little bit more functional than DNA. But our ultimate goal is proteins. They're the most functional. They're the business within the cells, and there are tiny machines. So that's why the major target for drug development for the past 50 years have been proteins. These are some types of enzymatic proteins. You'll see them in your research papers, and you can also search it up as well. But like it's good to have some idea of these, right? Kinases, phosphorylate. Do you guys know what phosphorylation is? Okay, we'll talk about it right now. Phosphatases dephosphorylate, they reverse phosphorylation. Proteases, we know about these. Molecular scissors. Ligases, I think we discussed a little bit in O levels, but it was extension content, so I don't know if you guys have gone through. Like glue things together, like in genetic technology. Tika. Transferases, they transfer functional groups. DNAs and RNAs, they cleave. DNA and RNA cleave basically means break down or like a change in its structure. And helicases also have a similar function. They remodel DNA and RNA strands. So when we talk about like when we learned that we're splitting the DNA strands when we're doing the transcription, right? We template strand and the non-template strand. This is by DNAs and helicase. Basic steps of translation. I hope you guys have gone through this as well. I don't know why I keep on repeating gone through it, but like originally when I was teaching this, many kids had not gone through it. So initiation, it's the ribosome gets together with the mRNA and the first tRNA comes in, Tika, and then the tRNA brings its amino acid. Two tRNA fit in the ribosome at once. These are ribosomal units. It forms one ribosome, right? And then when they're side by side, there's a bond that forms between them, peptide bond, and this repeats multiple times until we have a strand, polypeptide strand. That's our protein. Elongation, and then termination is when we're done. Tika, and then we've looked at this already, right? So there's the amino acid acceptor side, the attachment site, and then there's the anticodon. Do I need to go through the anticodons? You guys got it? It's like complementary to the codon and the mRNA. Do I need to for you because you've okay? So tRNA, this site that we're talking about, the anticodon, anti basically means the opposite, right? So when the codon comes in, it reads the it reads the codons, and the one that's opposite to it, the one that binds to it complementarily, that tRNA comes in carrying that amino acid, that specific amino acid. That's basically it. So just complementary binding. That's how it's like read basically. And then this is the basic structure of proteins, again part of AS, right? The primary structure, secondary structure, tertiary structure, and quaternary structure. So the primary structure is the main one that you guys will be looking at, but in your presentations you guys are going to pull up the quaternary structure, like back over here. This is the quaternary structure, and it's actually color coded. Like this is not like your proteins are not like rainbow colored, right? This is actually color coded to identify how much confidence we have in certain places, especially with you guys, because your genes are not as researched. So there are going to be certain places where they're like, I think it's white, where they're like not sure, not as sure if it's actually present there or not. But according to certain papers, there's a possibility. And then we know that secondary structure get extra bonds like ionic bond, hydrophobic interactions. They form tertiary structures. And then quaternary is just the interaction between different polypeptides. So from here to here, we're working with one polypeptide. And then with quaternary, we're working with more than one, like in hemoglobin. No, it's good. I remember my teacher used to like say some quirky remark whenever the students would yawn. I was like, yeah, I'm also really tired. Okay, now we're gonna get into our last topic of this cellular biology, which is post translational modifications. The sleep is getting to me. Okay, there are more than 300 different types. I'm not gonna go through all of them. I'll just name the main ones. Okay. It's usually the addition of small proteins or functional groups. So when we're talking about like when we learned in O levels, we talked about the wound healing cascade, right? Fibrinogen becomes activates this into this, and then fibroblast. I don't even remember. Yeah, thrombin, something like that. So that's actually the way that it's activated is that we're adding certain functional groups or we're adding smaller proteins to it. Okay? Modifications can be to fine tune or tweak the protein function or completely turn off or on and off protein activity. So you guys just recently went through in the same chapter Golgi bodies when we transport proteins for final modification in the Golgi bodies, Golgi apparatus. This is what we're talking about. It's getting modified by these PTMs, just its short form. It's carried out by other proteins and it's also reversible by other proteins. So this is the main one, phosphorylation, which you mentioned earlier. It's the addition of a phosphate group, like phosphate PO4, right? It's reversible, and it's carried out by kinases and reversed by phosphatases. All right. And PO4 is such a high energy molecule that it can cause the change in the protein's shape. So it can mask or expose certain areas of interaction between certain proteins. Like you can think about like in a very basic example, when we remove a non-competitive inhibitor, sometimes the active site can revert to its original space, so substrate can be attached, right? It can alter hydrophobicity, TK, and it can provide docking sites for protein partners. Basically, the same thing as the first point. It changes the shape so that proteins can bind together, like transcription factors, basically, right? The highly specific binding. And that's it for cellular biology. Do we have any questions before we move on?